Welcome to Parallax by Ankur Kalra, a podcast produced by Radcliffe Cardiology to bring you a new angle of all things cardiology and the best from the US Cardiology Review. Published every second Monday, Ankur Kalra, MD, interventional cardiologist at the Cleveland Clinic in Ohio, USA, speaks with legendary cardiologists, reviews late-breaking trials, and interviews authors of our latest and best US Cardiology Review articles. We call them #audio articles. Parallax is the effect whereby the position or direction of an object appears to differ when viewed from different positions. So this podcast is your fix of reliable updates on all things cardiology by someone from a non-traditional background who is always looking at the industry from a new angle. Now, here's your host, Ankur Kalra, MD.
Hello, everyone. We have with us a very esteemed guest, actually a couple guests. My first guest needs no introduction. He is a luminary in the field of interventional cardiology and has published seminal work in the field that has transformed the way we practice and take care of patients in the cath lab. It's my pleasure to introduce our listeners to Dr. Brilakis. Dr. Manos Brilakis is the director of complex interventions at my alma mater, at the Minneapolis Heart Institute in Minneapolis, Minnesota. And with him is his fellow and my friend Mike Megali, who's the first author of the paper which was published in US Cardiology Review, issue 13.1. The paper is titled Role of Drug-Coated Balloons in Small Vessel Coronary Artery Disease. Manos, Mike, welcome.
Thanks for having us. Thank you, Ankur. Thank you for inviting us.
No, it's my pleasure. So I'm going to begin the podcast by requesting you, Manos, to talk to our listeners a bit about how you define small vessel coronary artery disease in the cath lab.
That's a great question. First of all, again, thanks for putting this together, and thanks again for the invitation. The small vessel has to have some degree subjectivity because, as you know, it's sometimes hard to measure exactly if it's 2.0 or 2.5. But for many studies, and I think in the cath lab, 2.5 millimeters is the usual cutoff. We say if it's 2.5 or less, it's small. I mean, it's the 2.0 that really gets us going, or smaller vessels. So it's not 100% accurate, and we don't do QCA, obviously, but a 2.5 or less is a small vessel for most practical purposes.
Okay, and what percentage of interventions you think are performed in such vessels? Like in your practice and overall, if you look at the national trends, what percentage of coronary interventions would fall under the category of small vessel coronary artery disease interventions?
I think my impression is it's more in the 20% range. I know there are some national estimates that even half of them can be small. But I think about 1 in 5 cases, the vessel is going to be really small. What is useful to define small, understand a little better, is also to do intravascular imaging that gives you actually a quantitative assessment. But I would say 1 in 5 cases we do are in small vessels. If you look at vessels 2 or less, that's a little less than that, maybe 10%.
Sure. Now, in the paper, you've very eloquently described, some of the traditional risk factors which are associated or commonly associated with small vessel coronary artery disease, and those include old age, diabetes, peripheral arterial occlusive disease, and even lesions which are longer in length, tend to have an association with small vessel disease. My next question surrounds management. When it relates to small vessel disease, just walk us through some of the historical perspective, with regard to both balloon angioplasty and bare metal stenting for management of such lesions.
Sure. I guess I'll do the historical part since I'm the older guy, and then I'll have Mike do the more contemporary part. But the treatment of small vessel disease has been and continues to be a challenge. The stenosis rate with balloon angioplasty alone are very high, 50% or more. These are small vessels. Obviously, there is less room for recoil, and then there's less room for stenting, for forming neointima. And even for the drug-eluting stent studies, smaller vessel size, as well as lesion length and stent length, have been associated with higher stenosis rate. So the challenge is with balloon angioplasty that these are small vessels and they have high recoil. And the challenge with bare metal stents is that even a small amount of new intima may be all that is needed to make them occluded. But that's also true for drug-eluting stents, although they are much more effective in reducing the amount of new intima. There is still the possibility of restenosis, because even a 0.5 millimeter of new intima, that wouldn't be a big deal in a 4.0 vessel, is much bigger deal in a smaller vessel. I think also we're seeing this a little bit more because we have a more complex disease that comes to the cath lab. We like to say we don't see the type A lesions anymore, but we see a lot of people with diabetes. Diabetes prevalence is increasing, and I think the group that we quite often see this are diabetics, especially women with this very small diffuse disease vessels. So it is a real clinical problem. We do see this most days in the cath lab, and it is a dilemma what to do, which I will let Mike describe, but the issue is with balloon and long, which used to be an option. The results are usually not as good. Bare metal stents, I think, are a no-no for these lesions for many reasons, but specifically the high rates of in-stent neointima formation. Then drug-eluting stents is what we often do those days in the United States, since we do not have clinical access to the drug-coated balloons, although we've done a few cases of label where we actually take the drug-coated balloon in the coronary, but that's not the best way to go.
Sure. Thank you for sharing that with us. Mike, if you want to talk to our listeners a little bit about, the role of fourth generation, and then perhaps both you and Manos can comment on, the current second generation drug-eluting stents for, their role in the management of small vessel coronary artery disease.
Yeah, of course, as Dr. Rolakis mentioned, the use of balloon angioplasty and bare metal stents are associated with fairly high risk of restenosis, and there's been reported in multiple studies. And when the first generation drug-eluting stents came to the market, this reduced the ISR rate very well, and eventually the decrease of the ISR rates led to better clinical outcomes. However, they were still associated with higher restenosis, and of course, with the first generation drug-eluting stents, there were higher incidence of stent thrombosis as well. And this has been seen in some registries and some studies reporting about 6.5% restenosis rate in about a year, and this was still considered very high. And when the second generation drug-eluting stents came up to like the Resolute Onyx at 2 millimeters, and drug-eluting stents that were designed specifically for small vessels, this also further decreased the ISR rates, but because of the inherent limitation of the small vessels, because they cannot accommodate a certain amount of new intimal growth, the second generation drug-eluting stents were associated with still a pretty high risk of restenosis. And we could see that in the SPIRIT registry of about 5.1% restenosis rate and in 1 year. And when the Science was used, the Science registry was about 3.1%. So that's when it came to the idea of using drug-coated balloons in small vessels became very appealing.
Sure. No.
Say one more thing at this point. Although these numbers actually, to me, these numbers sound actually great. 6%, 3%, that's actually amazing, to be honest, for small vessels or any vessel for that matter.
Sure.
I think those numbers are probably affected, and in reality, what we're seeing is much more than that.
Yes, I mean, I think if you look at the overall percutaneous coronary interventions, I think the second generation drug-eluting stents have sort of become, the gold standard, right, Manos? I mean, in our daily practice, I mean, almost all interventions are either performed with everolimus-eluting or zotolollimus-eluting stents, which are our current generation, drug-eluting stents. And we could talk a little bit more about the polymer that is associated with stents. I know there is great utilization, at least in our cath lab, of the biodegradable polymer in the everolimus-eluting platinum chromium stent. And one would have imagined that with the introduction of 2O stents on the shelf, that they would be able to cater to this problem more efficiently, but sounds like it continues to remain a vexing problem. Am I right in that assessment?
Yeah, I think, even though the new generation, they have some promise, maybe thinner straps, or Cyrus coming up now. So there is a new development, but I think you're right. We didn't really see that much difference in terms of restenosis and reocclusion rates.
Okay. Now let's just get on to the topic of discussion, which is the use of drug-coated balloons. I know you brought that up earlier in the discussion, Manos. And to our listeners, I'd really urge you to take a look at the paper, particularly Table 1, which basically enlists a list of randomized control trials studying the use of drug-coated balloons in small vessel coronary artery disease. And we have Manos and Mike talk to us about the use of drug-coated balloons in small vessel disease. So why don't I request you, Manos, to begin the discussion, and maybe Mike could add on to it.
Sure, absolutely. So this paper, actually, the table has the data on the randomized control trials. Mike did another nice paper on this with a meta-analysis that also includes the observational studies, which there are several of those as well. But if we look at the meta-analysis that compares essentially drug-coated balloons with drug-eluting stents, except for one of the studies, the one by Funatsu that showed the comparison with uncoated balloon angioplasty, essentially the outcomes were similar in terms of target vessel revascularization, major adverse events, binary stenosis. So in essentially the 4 of the 5, which were comparing with drug-eluting stents, and 2 of them were with the Taxu stent, which is no longer being used, and the other 2 were with the everolimus and the zotolollimus stent, so the contemporary platforms we're using. But in both of those examples, the clinical events, both angiographic as well as TLR, TVF, MACE, all of those events were similar in the 2 groups. There was the one paper that the one study that compared with balloon angioplasty, and in that case, as expected, it did show lower stenosis with drug-coated balloons, although the overall TLR was numerically but didn't reach statistically significance. So the message here is that the drug-coated balloons are better than standard balloons in small vessels, but they're about the same as drug-eluting stents, with the advantage that we can talk more down the line, that you don't have the metal platform staying inside the vessel, which can be pretty challenging to treat if there's recurrent stenosis, more layers of metal in the small vessel, something we really don't want to see.
Sure. And this is a question which has come to my mind on several occasions. I'm sure it's come to others' minds as well, or at least I would hope so. Is what is the exact mechanism of action for preventing restenosis once you use a drug-coated balloon? I understand that there is abluminal delivery of the drug, which coats the surface of the balloon, into the vessel and the lesion. And if you can maybe delve a little bit more into the mechanism of how long does the drug stay in the vessel, and what is the concept behind treating such lesions with just a drug-coated balloon. I understand that the idea is to not keep the metal, because as we all know, that has long-term implications with regard to instant restenosis, particularly when the acute lumen gain is not significant. So if you can discuss a little bit more about the mechanism, I think that'll be very interesting for our listeners.
This is a very, very interesting question, Ankur. And This goes back to when the drug-coated balloons were first even thought about or invented, and that was around like 2003. And I would say that the studies in that was even preceded the drug-eluting stents, and the idea was to deliver the drug to the vessel wall by any mechanism and avoid putting in a metal in there. And then the discovery of drug-eluting stents and using the stents to deliver the drug, all this went contemporary with the development of, is there another way to have a stent-free delivery system, and use that delivery system to put the drug, the anti-proliferative drug, into the vessel wall without leaving a metal behind. And the drug-coated balloons are, like I'd say, an ingenious technology. They use the regular balloons, angioplasty balloons, and we mount the drug using an excipient. So the drug sticks to the balloon, and then we deliver the balloon to the lesion that is well prepared. And when we inflate the balloon, the drug actually is delivered to the vessel wall, and it is kept in the deepest layers of the vessels and does not stay in the endothelium, unlike some of the drug-eluting stents. So that allows endothelialization very well, but the drug stays in the vessel wall. And I would say the half-life and the duration of the drug being in the vessel wall, this depends on so many things. Excipient is one of them, how prepared the lesion was, and the manufacturing process of the drug-coated balloon, which is different among all the manufacturers. But we can say that if the drug uses paclitaxel, the current excipients used in the second generation drug-coated balloons, the drug stays for about 2 months in the vessel. And the difference between the drug-coated balloon and the drug-eluting stent is the drug-eluting stent depends on slow and consistent delivery of the drug in a very low dose, while the drug-coated balloon depends on an initial high dose of delivery to the vessel wall that degrades with time. And as we know, the restenosis process is exaggerated in the earlier phase of after the procedure, then that might make sense that while using the drug-coated balloon might help reducing the restenosis a little bit. In some opinions, that restenosis can take up to 6 months, and that's why we need continuous delivery of the drug, and that's why drug-eluting stents might be associated with a little bit more better angiographic outcomes. But the idea here is if we leave no metal, this finally ends up in same MACE at follow-up.
Thanks, Mike, for going through that mechanism. That's an excellent explanation. And, I think to complement that explanation, I'd like to guide our listeners to, or direct our listeners to figure 1 in the manuscript, which is a very well put together figure, which goes over schematically the mechanism of action for drug-coated balloons. Mike talked to us about, them being smaller in profile, promoting plaque reduction and stabilization, vascular healing, with the caveat then that there is a lower risk of restenosis with use of shorter duration of dual antiplatelet therapy. Let me ask you this question, and this may be a provocative question, but the question is, and Manos, you sort of alluded to this being off-label in use. It seems like we've decent data, right? If you look at Table 1, you've these randomized control trials. Granted that the denominator is not a significant one, or at least the ones that we're used to in the cardiovascular medicine space. But there is at least safety and efficacy data, if not longer-term follow-up. Why haven't these balloons made it to our shelves for commercial use?
So good point. There's clearly a clinical need, as you pointed out. There is a clinical need for, I would say, mainly for stent restenosis, because right now when stents or stenosis, especially with multiple layers, the next step is brachytherapy. And there's also a clinical need for small vessels and potentially some smaller subgroups like side branches or bifurcations, etc. It's clearly clinical need. However, those are regulated as a medication, and it's not an easy approval from the FDA. I know of several attempts to put the studies together to bring this for clinical use, but because of the amount of data and the studies and the cost that will be required, I think, versus the anticipated proceeds from the stents, I think that has been a challenge for the companies to bring this forward to clinical use. So hopefully it will come in the future. There's, again, as you said, a clinical need, but I think it has to do more of a business decision rather than the clinical part of this.
Do you use them in your practice off-label? I mean, is there at least, under special circumstance for patients, if you've expended all other options for revascularization, is there any window of opportunity for using these using these balloons in clinical practice?
It is extremely challenging. So we have used them, but let's not forget these are peripheral balloons. As small as like 4.0, I think by 20 or 30. So if these are big balloons, extremely bulky, these are very hard to advance over the 014 guide wire.
Sure.
So practically, you can only get them to like a proximal part of a coronary, or get them through a large saphenous vein graft. And there's not even it's never going to happen to get it down to a small vessel. So for small vessel disease, it's not an option. However, for stent stenosis, for larger vessels or saphenous vein grafts, we've done it off-label. We had to use a lot of techniques and tips and tricks to improve support and get it down. So it was not a fun procedure, but it can be done. Once again, it's not practical, and of course, we don't know about the dose you actually deliver. If it takes you 1 minute to deliver, 2 minutes to deliver the stent, the balloon to the target site, did the drug come off the balloon? I mean, there are many, many limitations. So can we do it? Yes. In reality, though, it's a very select patients, and these are very cumbersome procedures to them.
Sure. I'm going to use the opportunity of having you on the podcast, Manos, to just go over with us the technical aspects of, technical aspects of delivering these balloons, down large vessels. I mean, what are some of the tips and tricks that you utilize in the cardiac catheterization laboratory for, making sure that you get by these procedures? I think that'll be something which the listeners would love to listen from you.
Sure. It's the main things are the same for delivery of anything big and challenging delivery device. So first of all, you need to go with big guides. So 8 French is standard. And you want to be femoral access, you have better support. You want to use, in some vein grafts, we actually used glide wires, larger profile, like thicker wires. But if you use the standard 014, then we want to be as deeply engaged as possible. So if it's a vein graft, we deep seat the guide all the way to the lesion, essentially. If it's a coronary, let's say right coronary, you want to deep seat the guide again, almost all the way down to the vessel. A lot of strong support. So the 2-hand technique, one hand pushes the guide forward, the other hand advances the balloon. And then a lot of patience to be able to do it. And of course, before we do any of that, we want to predilate the lesion as much as possible. If it's in stent stenosis, which most of those are in stent stenosis, this will not go unless you've done big balloons 3/4. You really expanded the vessel very, very well, and everything is flying down. So strong guide support, strong guide wire, sometimes things like the Grand Slam, the wiggle wire, Ironman, very, very stiff wires, strong support. Then excellent lesion preparation with balloons or modified balloons, just make sure it's as expanded as possible. And then obviously we get the smallest and shortest balloon most of the time, which are the 4 the shortest potential length. So doing all this combination is still difficult, but it works for most cases. And
is there any special anticoagulation strategy that you use, or would you just do it on, intravenous unfractionated heparin targeting an activated clotting time over 250 seconds, or do you want to keep it to over 300 seconds? What is your practice in the lab?
Yeah, we have very few of those, but we usually go standard anticoagulation, 250 to 300 for most of the time.
Okay, terrific. Well, thanks for going over those steps. I'm sure our listeners would take that as a treat, coming from you. in conclusion to the podcast, what are some of the take-home messages you'd like the listeners to take out of this discussion and also the paper? And thank you for bringing up the meta-analysis that Mike put together, which I think is an important addition to the literature. Maybe you can go over some of the take-home points or concluding messages, from this podcast and from the paper that you published in US Cardiology Review.
Right, so I will say three things. The first thing is that small vessels remain a problem. Despite what we have, the second generation drug-eluting stents, I think you still get in clinical practice a non-insignificant amount of restenosis. Second point is that although drug-eluting stents is all we have in the United States, drug-coated balloons seem to have similar outcomes, and it would be preferred in most cases if we had them, given the lack of a metal frame into the coronary artery. And third, building on the second, that it can be done. Drug-coated balloons can be done in very selected large vessels, but the biggest thing is the need for finally getting a DCB in the coronary market in the United States.
Excellent. Thanks for the excellent summary of our discussion there, Manos. I'd like to thank both you and Mike for making the time to come and talk to us and share both historical perspective as well as new data and practical tips and clinical application of this technology. Thanks again for your time.
Hey, thanks for having us.
Thank you.
Dear cardiologists, we want to make this podcast about you and for you. So please email us your critical thoughts, comments and questions at podcast@radcliffe-group.com and visit uscjournal.com for more information. You can also follow us on Twitter, LinkedIn, Facebook and Instagram at Radcliffe Cardiology for daily updates. Join thousands of cardiologists and become a Radcliffean by registering to radcliffecardiology.com. You will receive regular newsletters and gain access to hundreds of expert interviews, educational webinars, clinical cases, and peer-reviewed articles from our 6 medical review journals on general cardiology, interventional cardiology, arrhythmia and electrophysiology, cardiac failure, and vascular and endovascular surgery. Thank you.